Don't worry, none of you are 70 and 80 years old, nothing's going to happen to you. Okay? What do you mean, inshallah? Nothing's going to happen to you. Okay, so relax with it. Of course. No, inshallah in the Arabic world means something else. Okay? Oh yeah, now I understand. It's not going to happen to you. And inshallah means, khalas, it will never happen. Okay, good point. Right. So today we're going to start off with blood, and we are going to be focusing on erythropoiesis. Why? As an immunologist teaching you this, I have no idea, but we are going to do that anyway. So start off. What do you understand by erythropoiesis? Okay, let's not mumble, let's be focused. Development of blood, totally wrong. If you're not considering 0.1% in the equation. 99. So blood, what is blood? Let's start by that. No, definitely not. Good. Blood is a connective tissue. Okay? A connective tissue that has got two components. One is the liquid part of it, and the other is the cellular part. In the cellular part, you've got the red blood cells, white blood cells, and the platelets. Okay? Although platelets and RBCs do not conform to what you call a cell because it is not nucleated. In the liquid portion of it, you've got the plasma, and within the plasma, you have plasma, pardon my handwriting, proteins, then you've got nutrients in it, which can be your glucose, amino acids, lipids in terms of cholesterol, HDL, LDL, etc. Then you've got ions in it, sodium, potassium, bicarb, etc. And of course, you have CO2, urea, and other amine waste products that can come with. Okay, broken down products. Plus, of course, the drugs that you try to take, whether they're water soluble, lipid soluble, come in. Now, if you look at the division of it, the cellular component approximately is 45%, whereas this whole section is around 55%. If you take just in terms of volume measures, and within this, within the plasma proteins, you have again three fractions. You've got albumin, which is by the most abundant part of it. Then you've got globulins and fibrinogens. Fibrinogens, along with the platelets, are important for hemostasis. Hemostasis is what? Clotting. Okay, which is different from homeostasis. Homeostasis is maintenance of the internal environment. Hemostasis is coming back to the clotting. Now, amongst this cellular component, RBC is the most dominant type, 99.1% of this whole 45% is these cells. Okay? The rest of the cells, WBCs and platelets, come around 0.9 or 1%. Okay? Now that is how much of an RBC you have. So when somebody says, what is blood, and they just say RBC, yes, you're overlooking this part and you're overlooking this part. Okay? Now, I forgot even my first question. Erythropoiesis, right? So now, what is erythropoiesis? Erythropoiesis is, from this whole component, just the formation of one part of it, which is your RBC. Yes, we do agree, by far it is the most common and the greatest in terms of proportion, but that does not mean erythropoiesis means formation of blood. Blood is really much more than just that. Okay. Now, today that is going to be the focus of our talk, and it's primarily the formation of RBCs. Where do RBCs come from? RBCs are coming from bone marrow. And why do you think all of these cells actually are coming from the bone marrow? Why do you think they're coming from the bone marrow? What is there in the bone marrow that is responsible for all of these cell types? Good. So your bone marrow is a very conserved environment with a lot of factors. So there are two kinds of things you need to remember, right? Factors that are produced can give rise to something called as proliferation or differentiation. Proliferation simply means that if that factor is there, that factor could be anything. It could be a hormone, it could be a cytokine, it could be any product that is made in that particular environment. That factor can either just act on the surface of a cell or inside the cell, depending where the receptor is, and induce multiplication of the cell type. That is simple cell division. Or it can act on the cell and ask the cell to change its behavior. Okay? So factors could be cytokines, hormones, etc. The bone marrow is a particular environment where you have a lot of the factors that sustain or maintain one population of cells, and these are called as your pluripotent stem cells. Okay? If these pluripotent stem cells are going to give rise to the cells that are found in the blood, you end up adding an agent. Pluripotent hematopoietic stem cells, meaning they're going to give rise to hematopoiesis. Poiesis means development of or giving rise to. Hemato primarily means coming from the blood. So they give rise to the cells of the blood. Why do I say that you have pluripotent stem cells? Because these stem cells can actually also give rise to other organ types. You can take stem cells from the bone marrow, and if you differentiate them in a particular manner, you can actually make a liver, a neuron, any other cell type. Okay? But for those stem cells that can give rise to the cells of the blood, whether they are RBC, WBC, or platelets, you call them pluripotent hematopoietic stem cells. Okay? Does that make sense? So if you look at the bigger picture, you have your hematopoietic stem cells that have a capacity to renew themselves. Okay? And that capacity of cell renewal comes from the factors which are produced within the bone marrow for self-proliferation. Okay? Now when we talk about the pluripotent hematopoietic stem cell becoming an RBC or a WBC, it will then have another set of signaling pathways that will divert it. Instead of just remaining in a cycle of self-division, they will start differentiation, start becoming different cell types. They will gain different attributes and lose previous attributes. Okay? Now, from them, you end up having common myeloid progenitors or common lymphoid progenitors. Okay? So this is the process of differentiation kicking in. The common lymphoid progenitors can only further go through processes of differentiation to give rise to the lymphoid cells, which are the B lymphocytes or the T lymphocytes, right? Or they can give rise to another, so the common myeloid progenitors will eventually give rise to the RBCs or the platelets. Whereas you can have the granulocyte monocyte progenitors, which is another lineage coming out of here, which gives rise to your cells which are granulocytes, which are neutrophils, your macrophages, eosinophils, including mast cells. Okay. Now, today's talk is going to focus starting from the pluripotent hematopoietic stem cells, going to the common myeloid progenitor, eventually ending up into the formation of RBCs. This is a bigger picture. As we go along, we'll see more in details how things happen. Now, I'm going to take a small pause here because we need to go back into a process of reflection. Now, stem cells. What do you think? That's fine. I don't mind being green. Okay. Stem cells. What is a common important factor that stem cells need to have? Okay, so stem cells need to have DNA. What else do they need to have? Okay. Receptors. Receptors, good. So a cell needs to have a DNA in the nucleus, and on the surface, or in the cytoplasm, or in the nucleus, depending on its locality, it needs to have receptors that will respond to the external stimuli, right? What else does a cell need? Sorry? Excellent. It needs to have a source of energy, and that source of energy, for all we know, is supplied majority by mitochondria. Excellent. So, because this cell is going to give rise to protein synthesis, it will also need to have ribosomes to make proteins, and the DNA is primarily going to be coding for those protein synthesis to take place, right? And the ribosomes could be free ribosomes in the cytoplasm or associated with something called as the endoplasmic reticulum, depending on the kind of protein that's going to be synthesized. Are we all fine with this? What else do you think this cell needs? Transcription factors. So transcription factors will be downstream of the receptors. So signal will come to the receptor. The receptor will signal, giving rise to some sort of transcription factors. That will tell the DNA to make particular mRNA, and therefore the protein of interest, or the particular protein that then needs to be made. Are we fine with this? Now, opposing to this, what do you have in an RBC? Hemoglobin. So, you have an RBC, and all that you have in that is hemoglobin. Anything else? Okay. Let's not make it that simple. Okay. We do need a bilipid layer. Okay. We do need some cytoskeletal proteins, right, to keep it in its shape. We may also need some amount of sodium-potassium pumps to maintain it in there. And we need, over here inside, the RBC does require energy to maintain structures inside it, and including the sodium-potassium ATPase pumping. And that energy is going to come from where? Mitochondria. No mitochondria. Make up your mind, guys. Surrounding environment. So ATPs can just diffuse in. That could be a good idea. But how many of you lend each other money this often? I guess not. So let's say that we can put something called as glycolysis, which is cytoplasmic in nature, right? And apart from that, we have another process which is called hexose monophosphate shunt. Now, the ATP, so think about it, right? Mitochondria have electron transport chain, and they are the highest consumers of oxygen. The function of this cell is to transport oxygen, right? So if your parents give you money to go buy pizza for a guest that just popped in, and if you on your way back finish the pizza, right, you're not going to be loved by your parents. So if this cell is going to suck up all the oxygen, what is it going to transport, right? So that's why this cell does not have any mitochondria. Does it have a DNA or a nucleus? No. So if you don't have a DNA, you don't have a nucleus, therefore you will not be having mRNA, therefore you will not be able to make new proteins, therefore you don't need ribosomes and endoplasmic reticulum, right? So a cell that is not going to be doing any protein synthesis will not require all the machinery required for protein synthesis, right? And therefore, will this survive very long? No. After 120 days, you'd have the good and the bad and the ugly, and you'll have the macrophages finishing it off, right? So, from this stage, where a cell is a pluripotent hematopoietic stem cell, over different processes, we need to end up making a cell That is primarily this. Okay? So now you know your start point, you know your end point, and we're going to be talking a lot about what is going to happen in the middle of that process. Yeah?
What about anaerobic respiration?
Sorry?
Anaerobic respiration.
Glycolysis?
Yeah, so
HNP shunt. That's what's going to be the major source of ATP generation here. By the way, ATPs outside the cell are a big no-no. Okay? They actually activate the immune system and cause inflammation. Okay? Similar to DNA, outside the cell is a big no-no. Your immune system recognizes it as an antigen and gets activated against it. Now, I'm just going to run through some crappy slides because that's just information. Embryonic stages, erythropoiesis kicks in very early. You have 3 stages. You've got the mesoblastic stage, hepatic stage, and the myeloid. Mesoblastic is as early as the 3rd week, and primarily the RBCs are nucleated at that stage, and they develop from the yolk sac and the mesothelial layers of the placenta. Later on, at 6 weeks, the liver takes over, and the spleen and the lymphoid, and later at 3 months onwards, it moves to be the myeloid stage. Now, bear in mind all of this. You definitely, as adults, do not have the yolk sac anymore, okay? And you don't have the mesothelial layers of the placenta. You know, that was the, whatever is left is just a belly button as a remnant of the placenta in you. But what you do have is the liver, spleen, and the lymphoid tissue. Why do I put this in perspective? Is because in adults, yes, all of your RBCs are almost exclusively made by the red bone marrow, but you can have situations in adults where the bone marrow fails to function, and the machinery that existed in your primitive state when you were still in the embryonic stages of development, they kick in, and the liver and the spleen start making RBCs. That is called as extramedullary hematopoiesis. Hematopoiesis is RBC production. Extramedullary means it is outside of the bone marrow, the medullary canal of the bone marrow. So these things are actually seen in adults as well. Interestingly enough, your liver and the spleen are also the main sites where your RBCs, by the time they have aged to 120 days, are actually broken down. Majority of the breaking down is taking place in the spleen. Minority takes place in the liver as well. Okay? Postnatally, we know that it's all the long bones. I'm not going to waste time over this. You can just read through these slides. And of course, as time progresses, the bone marrow that you have, which is the red marrow, starts slowly getting converted, and the space is taken up by fat cells, becomes what you call as the yellow or the white marrow. And by the time, I'm somewhere here in this lineage, you guys should be somewhere here. So yeah, you got much more in the sternum, femur, vertebrae, whereas for me it's not functioning anymore. Only the flat bones in my body are worth anything now. And by the time I'm 70, soon, by the time I'm retiring, my femur will give away as well. Okay. Now, how many of you know the structure of hemoglobin? Okay, question. How many of you don't know the structure of hemoglobin? You know, I like more votings. Good. Okay. We'll just, because that is not the scope of our talk, but it's still relative to our discussion, I'll just go quickly over. Basically, you have 2 alpha chains and 2 beta chains that are linked to each other, and within them, you have over here a heme group. Now, what the heme group looks like is a protoporphyrin ring with ferrostate of iron, Fe plus 2 in the center. As long as iron is in its ferrostate, Fe plus 2, it is actually able to bind to oxygen molecules. The moment it itself gets oxidized and becomes ferric, Fe plus 3, it is not able to bind to oxygen anymore. that is called as methemoglobin. Okay, so agents which strongly oxidize, they convert it into that, and you lose the oxygen carrying capacity of the blood that would come in. Now, why are we talking about this? Is because your RBC primarily that is over there, like he very correctly stated, is hemoglobin. It is a sac full of hemoglobin, and you have around 280 million molecules of hemoglobin in just one RBC. So that is a lot of hemoglobin that is focused, put down in one cell type. And it's because of the hemoglobin that is there. You remember earlier we talked about blood as being 55% liquid and 45% cells. So if your blood was only liquid, its hemoglobin, its oxygen carrying capacity would be very low. Okay, you know oxygen can be dissolved in liquids? Yes, no, maybe?
Yes, yes.
Show me an evidence.
Water under sea.
Water under sea, therefore. All sea life is dependent on the oxygen that is dissolved in the seawater, and therefore they survive. So your blood by itself, the liquid part of it, the plasma can carry oxygen. But its capacity would be very low. The volume of blood you would require would be 70 times more. An average person has got 5 to 6 liters in circulation. To compensate, you would have 350 liters required to supply enough. So hemoglobin actually that is there increases the capacity by 70-fold and therefore helps in carrying out this particular functionality. Now, again, going back to this image, we need to start from a pluripotent cell type to a cell that is primarily filled with hemoglobin and enzymes required for glycolysis, which provides 80% to 90% of the energy, and hexose monophosphate shunt, which provides another 10 to 20% of the energy required by that RBC. So your pluripotent hematopoietic stem cells eventually give rise to something called as colony-forming unit erythrocyte under the influence of things that we already discussed. Now, two things you need to understand. Your stem cells have the capacity to give rise to asymmetric cell division, and that's what these stem cells are doing on an average given day. Right now, the stem cells that are in your body are actually giving rise to more stem cells and giving rise to progenitors. Now, depending on the growth inducers and differentiation inducers, so the growth inducers will give rise to more stem cells, and your differentiation inducers will give rise to more progenitors. So this is what is happening on any given time point. Now, your blood is a very important part of your existence, and therefore you have mechanisms that need to be in place to ensure that it functions well. Okay? Why do I say that? Because all of us are prone to bleeding, right? Women particularly go through a menstrual cycle every month when they are coming of age, and men are prone to injury just by the default brain mechanistic aspects that they have. They just need to break a nose or something, or drive a car very rash. Thank you for demonstrating that. None of you saw that, but something happened in them. So by default, you are able to lose a lot of blood, and the important part is your body needs to have a mechanism to replace it very fast. Okay? To be able to do that, you need to have something called as symmetric cell division that kicks in, and this is primarily not going to be in from the, this is primarily going to be pushing in depending on whether you get growth inducers purely or differentiation inducers purely. So your stem cells, under the impact of erythropoietin after a huge blood loss due to either you donate or have an accident, pushes things towards making differentiated cell types, and the stem cell directly go into becoming progenitors. Alternatively, you can be exposed to irradiation of a part of your body and you lose your stem cells. And because of that loss, your stem cells can go into more symmetric division where they give rise to more stem cells because they need to simply repopulate the environment. But for healthy individuals on any given day, you're going through asymmetric cell divisions where you give rise to stem cell population and the progenitor populations. Now, erythropoiesis by default is split into two stages. You have an early stage and a late stage. The early stage is primarily focused on giving rise to committed cells that cannot change or cannot be pushed into any other lineage. Starts with the pluripotent hematopoietic stem cell giving rise to the myeloid progenitors, from where they give rise to burst forming unit, colony forming unit. After that, they move into late stage where they give rise to proerythroblast, polychromatophil erythroblast, orthochromatophil erythroblast, reticulocyte, and the erythrocyte. Now, too many words, too many names, but they're really very simple to understand. In the early stages, because they are still somewhere in the bone marrow and the stem cells, you are not able to differentiate between cells morphologically. What do I mean by that? The others would also. So think of this, right? All of you are morphologically different. I can look at you, I may not recall your names, pardon me for that, but I know your behavior, attributes, and your faces. I see you in the mall, I know you, I change my direction. Okay? Morphology. I know where you are and what you look like. That is morphology. So proerythroblast is going to be looking different than a polychromatophil, orthochromatophil, reticulocyte, and erythrocyte. So you look under a microscope and you can see they look different from each other. When we say that they are morphologically unidentifiable, meaning that I'll make, for example, a clone of him. Okay? The clone may have a different mindset. He may be more evil or less evil than he is. But on the outlook, they will have a similar morphological feature. I cannot differentiate between them. Okay? So these cells that are here are morphologically the same, but behaviorally they are different. Okay? So there's no way under a microscope you can differentiate between them. But how do you differentiate between them is when you take the pluripotent stem cells and you grow them in vitro cultures. When we do cell cultures outside the body, we put them in incubators and grow them. When you put with them different kind of factors, the growth factors or differentiating factors, they show different kind of behavior. Now, the burst forming unit, by name itself, you can identify that this gives rise to blast cells, clumps together. Okay? And colony forming units, they do not proliferate as fast, but they try to aggregate and clump together to form small different colonies. Now, of course, the first segment, all of it, is focused in the bone marrow, although they are early and late stages. And within the bone marrow, they take around 4 to 5 days to continue from a progenitor all the way to the orthochromatic erythroblast stage. However, outside the bone marrow, you still have the reticulocytes that would come out, and they would remain in the blood for around 1 to 2 or even up to 3 days till they actually mature into the erythrocyte. Of note is that all of this happens normally, and like I said, under states of stress where you have had blood loss, or you go to high altitude where RBC requirement suddenly increases, right? This process will be pushed more. So eventually, in the peripheral blood, you have majority of your cells as the mature erythrocytes at any given time point, and a few reticulocytes. But if you have a stress induced and more of this is formed, you will have what you call as reticulocytosis. Increase in the number of reticulocytes because of increased demand. Okay? And that by default means is under the influence of erythropoietin, because erythropoietin is the one that is pushing in this particular direction for proliferation and giving rise to erythrocytes. Does that make sense? Yeah.
Everything makes sense except the state between myeloid progenitor to proerythroblast.
Mm-hmm.
Can you
Oh, it's coming. Oh, there's so much more coming, don't worry. So you have pluripotent hematopoietic stem cells, and like we said, they need to have some level of self-renovative capacity. Their self-renovative capacity is primarily due to three things. You need stem cell factor, interleukin 6 and 11. All these three, when they signal, they give rise to more of pluripotent hematopoietic stem cells that by default are only 0.01% of the total cells found in the bone marrow. So they are minority with the capacity of self-renewal that's going on. After several stages, they end up coming out of the bone marrow as reticulocytes or then the RBCs. Now, some growth factors are known as common growth factors that are impacting all the precursor cell types. Stem cell factor, interleukin 6, granulocyte monocyte colony stimulating factor does this. Okay? Then you have another group of factors which are called lineage specific growth factors, meaning that these growth factors, once they are there, they will only promote the direction of formation of one cell type or the other and not any. Okay? For example, erythropoietin is a lineage specific growth factor that will give rise to RBCs only. Thrombopoietin is a lineage specific growth factor that will give rise to platelets, okay, or megakaryocytes that will then give rise to platelets. And granulocyte colony stimulating factor is going to push towards neutrophil development. Okay, do you get that? Now let's move to the erythropoiesis and its stages. Now you have the myeloid progenitor. The moment it comes under the influence of interleukin 3, which pushes it towards differentiation, stem cell factor that is going to support its proliferation, and erythropoietin, which allows maturation and blocks apoptosis. You will end up having a group of cells which you call as burst forming unit. Burst forming unit is only a convention that we look in vitro in slides outside the body. Inside the bone marrow, they all morphologically look the same. But when you take them out and you put them into culture, they give rise to blast cells in clusters. They have an ability to replicate very fast, proliferate very fast, and they form satellite clusters around core of the erythroid cells. All stem cells are CD34 positive. You should know that by now after human structure function and unit 2 body defense. Okay, so CD34 should be positive over here. CD45 is also positive over here. As they move on further, they still remain CD45 and maintain CD34, which is still a stem cell marker. Now, because they have gone through the differentiation, they do not require the signaling anymore for interleukin 3. Therefore, the way they shut off the signaling is just decreasing the receptor expression on the surface, right? They do not have the control of stopping the IL-3 being produced by the environment, right? You get what I'm saying? Just like you guys right now have no control over my blabbing. You can just shut your brain and go to Mars or Neptune or fairyland and stop the input coming to your brain, okay? So similarly, they can get rid of the receptor and block the signaling coming from the outside, and that's what they do. However, and they have low expression of CD71. What CD71 does is still a debatable aspect. They're still trying to find out its exact functionality. It's expressed, however, on these cells. From here, they move on and become colony-forming unit erythroids, where they're still CD45 positive, but lose CD34. They remain IL-3 negative and now start expressing more amounts of CD71. So basically, there's a small difference in signaling that is taking place to them, and the colony-forming unit erythroid is the last cell that you can say has stem cell-like properties because it's losing them by down-regulating or removing CD34 from its surface. Okay? Now, erythropoietin drives the direction of the myeloid progenitor for it to becoming a burst-forming unit, and then continues to do so for it to become a colony-forming unit. Okay, so this is what we were talking about, that these are lineage specific signals that come in. Okay, now we'll move to the stage of pro-erythroblasts. We can pause here a little, and if you have any questions, you can ask me. Yes.
Yeah, like you said,
I
the
still have.
last,
There's no timing here. Good.
remnants of stem cells is at CFUE, right? Colony forming unit.
Yeah.
Those are the last remnants of the stem cell.
So at that stage, it loses its stem cellness properties. Okay. Before that, it still retains CD34 positivity, which is more towards the stem cell. And if given enough pushes, it can go right and left. Okay. Now, let's move on to the late development stage, right? So you have the proerythroblast that becomes the basophil erythroblast, polychromatophil erythroblast, moving on to orthoreticular erythrocytes. Now, erythroblasts, what does that word tell you? What are blast cells? Blast cells are immature, but they have one very important property, and that is proliferation capacity. They can multiply and divide very fast. When you call something as pro-erythroblast, meaning this cell is pro towards developing, it will give rise to the erythroblast that will be developed. Now, how many of you recall H&E staining? You know what H&E staining is? Yeah. So let's get back to that part. What do you understand by H&E staining? Subhanallah.
It stains depending on the charge.
So this is a cell. Okay. What is inside the nucleus? DNA. Deoxyribonucleic acid. Okay. So anything inside, so this is full of DNA and some amount of RNA may be there. Okay. The cytoplasm is full of the cytoskeletal proteins, right? Now, what have I written down there? That's called cerebral atrophy, isn't it? DNA, finally. Now, if something is acidic, it will like to go towards the opposite, right? So, North South Pole, I will not give other examples. So, acids like to get entangled with basics. So, if you have a dye that is basic in its nature, okay, it will like to stain the nucleus more. So, the basic dye, the one that you see as deep blue purple, is the basic dye. Baso is basic, phil is to like. Okay? So basophil erythroblast has a cytoplasm as well that likes to take a deep purple stain. So the nuclei of any cell type will always be basophilic because they have nucleic acid, right? But you remember from white blood cells we talked about basophils. You remember them? Neutrophils, basophils, eosinophils. So a neutrophil does not take any dye. It will not take the deep purple dye or the pink dye. Okay? A basophil will have granules in its cytoplasm that like to take on the basophilic dye, and therefore they're called basophils. Eosinophils, if you recall, they had in their cytoplasm granules that like to take acidic dyes, right? Now it starts off with the basophil erythroblast having what in its cytoplasm? So there are no granules here. Granules were white blood cells. DNA is nuclear in its origin. So what can you expect in the cytoplasm? Remember, slow down. I know you guys are very smart. Slow. From DNA to protein, what needs to be made? Exactly. So the cytoplasm of these cells is going to be full of RNA and therefore the basophil. From there, you have something called as poly, means many, Chromatophil. Chrome, you know chrome plating, chrome's colors? You have those really cool cars that change their color because you're looking at it from a different angle, and then you don't know whether it was yours or somebody else's if everybody bought them. But anyway, those polychromatophil means that you have many colors. Now the color change is taking place from a basophil going towards eosinophilic. Okay? And in between, you have a stage where you have basophilic and eosinophilic staining coming together. Meaning, from RNA, you have started protein synthesis, but there is still a lot of RNA and still a lot of protein in this cell. Do you get what I'm saying? Is easy or just damaging your neurons? Yeah? Yeah, so you see, the cytoplasm of the cell, what does it stain? Pink, right? Eosinophilic in nature, because they are proteins primarily, right? They like to take up more of the acidic type. Now, when a cell is transitioning from being total RNA and losing the RNA and becoming more proteinaceous in nature, it will carry both colors. That's why you call it polychromatophil erythroblast. Okay? Then you have orthochromatic, meaning the colors are reducing again. Both of them change till you're reaching a stage of reticulocyte, where the cytoplasm is primarily full of protein and the RNA content is gone away, eventually giving rise to the erythrocytes. Yeah? It will have protein in the cytoplasm, but the proportion of RNA will be so high that it will dominate the staining. Okay, of course it has to have cytoskeletal proteins to maintain it. Do you get this point? Now, Alhamdulillah, I'm happy that there's no timer running, so I don't know how much I can teach. Oh, I can teach quite a lot. Good. So, is this part clear to all of you? So now you'll be able to understand not only the color, but relate it to their function. The important part is iron, right? Have you gone through iron metabolism?
Yes.
Oh, lovely.
So reticulocytes are just immature erythrocytes?
Yes, yes, almost. We'll discuss that. So iron metabolism, since you guys know, please guide me. From food. Food and diet. But then, oh, diet, not diet, sorry.
Taken as Fe3 plus.
Fe3 plus, Fe plus 2. Kumbhisnillah happens by the word of its Lord. Kumbhisnillah or something happens. Okay. Side. What happens to this then? Yeah, done. So the gut goes through, fine. Blood. What in the blood? Oh God. Transferrin. Shukr, alhamdulillah. But then,
Depends on where is it needed.
generally, where is it needed? Yeah, but today we are making it needed where? No RBC. Today we want it to go to the RBC, right? Of course, liver later, some other day when we talk about liver. Bone marrow, how will it go there? Blood.
Transfer binding to
Agree. Okay, interesting choices, interesting things. I agree with most of what you're trying to say. One important thing is, where were RBCs breaking down? Majority was the spleen, good. And in the spleen, what cell type is trying to help in the breakdown of the RBC?
Macrophages.
Interestingly enough, the macrophages that are in the spleen that break down the RBC, okay, and in the end, you have bilirubin being released outside, which is the broken down protoporphyrin ring that we discussed in the hemoglobin. Heme, alpha and beta chains are also cleaved off, released as amino acids for circulation and therefore reutilization. Iron, all of it is not released. Iron remains sequestered within the macrophages. Okay? This is an important point. And these macrophages, lo and behold, end up going to the bone marrow. Within the bone marrow, they give rise to what you call a small islands of erythropoiesis. These macrophages in the bone marrow are actually the source of iron. And around these macrophages, you have the erythroblastic stages taking place, where this iron is slowly being released to the cells, because eventually these cells need to end up making what? Hemoglobin. Okay? So when it starts on the basophil erythroblast, moving to polychromatophil erythroblast, that's where iron is typically being released. What else is important for these cell types is folate and B12. B12 and folate are required for what? Nucleic acids, right? And therefore they are more important for the basophil erythroblast stage and the polychromatophil, not so much for the ortho and the reticulum. Because this is where cell division still takes place and it is required, not at the later stage. And of course, the RNA production. Now, apart from that, the last part where you see the orthochromatic erythroblast converting into reticulocyte. Notice that the orthochromatic one right there still has a nucleus. The moment it becomes a reticulocyte, it is an anucleated cell. Where does the nucleus go? Macrophages eat up the nucleus. Okay? So the whole process is taking place around a central nidus of macrophages. Macrophages are the source of iron for these cell types as well, and for the last stage, they need to take away the nucleus as well. Right. Yeah?
How the iron from diet goes to macrophages?
No, dietary iron will also come directly to the bone marrow by the blood itself. But it is not enough for the RBCs because they're going into a rapid proliferative cycle. From there, you have transferrin that is primarily going to be carrying it, but you have other iron transport proteins that are basically ferroportin, which primarily transports it intracytoplasmically into the macrophages particularly. Okay? Now, so your proerythroblast, okay, is the cell type which has got a high nuclear to cytoplasmic ratio. You see the nucleus is really large, cytoplasm very scanty. Because there's no real function of this cell except it's an intermediary stage. Now, apart from this cell is able to go through 4 or 5 divisions. It has got very prominent nucleoli. I don't know how visible they are, but you see these darker areas, they're prominent nucleoli. And it has got small projections, these projections on the cytoplasm, which are ear-like membrane projections that it has. From this stage, it progresses very fast into becoming a basophil erythroblast. We already discussed that this has got high amounts of RNA, and a small amount of hemoglobin just starts to be produced over here and continues so when it is in the polychromatic erythroblast stage. Here, the way you are able to morphologically differentiate is that the nucleus, well, this picture is very bad, the slides are slightly better. It has sort of a checkerboard pattern in the nucleus of it. And that's why you're able to identify it as polychromatic. In addition, you will see that in the cytoplasm, it has got both basophilic and eosinophilic staining coming together. Gives more of a grayish hue to the cell rather than pink or blue. Okay? Now, then you go to the orthochromatic erythroblast. These are also called as the normoblast. Here the nucleus shrinks, and because the functionality of now the nuclear functionality is being reduced, more of these cells are making more hemoglobin, so it becomes more pinker in nature. And by the end of it, the nucleus is pushed to one side of the cell. Now, the moment this nucleus is pushed off to one side, it becomes enucleation. A process of enucleation kicks in where the nucleus is pushed out of the cell. What is not clear is whether the cell pushes the nucleus out or a macrophage eats it. Okay? These processes are still not clearly known, but macrophages phagocytose, and the nucleus moves out, and eventually it becomes a reticulocyte. Now, this is, and the macrophages primarily have a lot of an abundant enzyme called DNase 2. And DNase 2 is important because it is going to break down the nucleic acids required over there. Here you can see a nucleus being extruded out of the cell. Now, and here you can see that you have the cells being broken down by itself during that particular process. When you go to scanning electron micrograph, the nucleus that is being extruded or removed out of this cell at this stage has a very thin rim of cytoplasm and a membrane around it. And immediately next to it, you have a macrophage that usually likes to phagocytose and take it away. At this stage, the cell still has mitochondria in it. Okay? So the reticulocyte that you have has a cytoplasm, still has cytoplasmic organelles, quite a number of ribosomes, few mitochondria. And now these all organelles go through processes of breakdown through the proteasome. You remember what was a proteasome? Right? These organelles, the ribosomes and others, grow down through proteasomal degradation, and over the next 1 to 2 days, while the reticulocyte is circulating in the peripheral blood, it loses all of this, and certain cytoskeletal changes come in, and it ends up becoming what you call as a mature RBC. Mature RBC has got more cell membrane than the cytoplasmic content. Therefore, it has a high surface area to the volume ratio. That's why it's biconcave. Okay? And that's why it can have the flexibility to move on. At this stage, the reticulocyte primarily goes through a process of diapedesis, moves out of the bone marrow, ends up coming into the circulation. This diapedesis is quite similar to what you see in neutrophils when they move out of the blood vessel to go to the tissue in terms of infections. Now, is this clear to you guys all? Okay. Now, what regulates the production was going back to the erythropoiesis kicking in by erythropoietin. Where is erythropoietin made? Kidneys. Good. So erythropoietin is primarily a simple peptide hormone, and it's the interstitial renal cells that are primarily there in the peritubular areas, and that's where erythropoietin is coming out from. So they act as oxygen sensors. Now, the moment, of course, and they go back to the bone marrow and the cycle repeats. Now, erythropoietin as a hormone, and primarily the peritubular cells that recognize and act as oxygen sensors, they do not immediately kick in erythropoietin production. So you don't have preformed erythropoietin. You have preformed insulin. The moment you eat food, it is released. Erythropoietin needs to be synthesized and then released as it does. And the important point is that you need to have sustained hypoxia. Low oxygen tension that has to be sustained over at least 15 to 18 hours for erythropoietin levels to rise. And in some cells make a basal level of erythropoietin. In hypoxia, other cells join in and make more. So your erythropoietin levels maintain the RBC levels. Okay? Just like interleukin-7 levels maintain the T lymphocyte levels. You drop IL-7, T lymphocytes drop. You drop erythropoietin, RBCs drop. You raise erythropoietin, RBCs go up as well. Okay? And that's primarily it. So you can have defects in erythropoietin functionality, and you can have situations like polycythemia where you have a cancer of the precursor cells of the RBCs. They give rise to more RBCs, including reticulocytes. And because you simply have an increased RBC mass, you have decreased erythropoietin production. But still, that synthesis is out of control over there. This is what would happen in hypoxia, that you have decreased arterial oxygen saturation, pushing the kidneys to make more erythropoietin and then compensating for the whole loop. And anemia, I don't know, it's gone all the way there, would have a decreased cell mass and also increased the erythropoietin production per se. Okay, so 2-3 key take-home points. One is the function of the macrophages is crucial for RBC synthesis. Okay, people who have got defective macrophages are actually not able to make RBCs. Another important point is that these macrophages also provide the iron that is required in that area, in addition to getting away of the nucleus that's going to take place. And your erythropoietin is the key factor that's going to drive all of this. Okay? So I guess by the end of it, we'll stop right here. I was thinking of talking about erythropoietin signaling, but at 13:31, and your head's drooping away to one side, I would say that maybe we'll pause over there. So this slide, you can just maybe read over yourself because it's more molecular biology and signaling. Basically, it signals through JAK-STAT pathway. You need to go through it yourself. Please do spend time reading it. Don't waste an hour over it, but I would say at least waste 30 minutes of your lives reading about it because it's going to help you understand things better. Okay? I'll stop right here. You have any questions? We can go for them.